Hello folks, today is our last day of content and we are going to cover nucleotide metabolism. Here are the learning outcomes for the day. We're going to recognize the complex nature of the synthesis of nucleotides. We're going to compare some common substrates used for both purine and pyrimidine synthesis. We'll look a little bit into the regulation of each pathway, and particularly focus in on the two flux controlling steps. We're going to synthesize our knowledge of substrate and product availability within pathways with how regulation happens within those pathways. And lastly, we'll see how DNDPs, this is deoxynucleotide diphosphates, from NDPs, nucleotide diphosphates, and thus latter would be containing the ribose, not the deoxyribose. Our media scientist moment of the day is Hamed Haroon. He is a research associate at the University of Manchester, and he is a wheelchair user. Growing up, he wanted to be a physician, but he was told because of his disability that he couldn't do that. So as he was exploring science, he realized that actually science was another way to get into a field related to medicine, and he found himself actually more interested in the science side of medicine. So he got degrees in both medical physics and a PhD in MRI analysis, magnetic resonance imaging. And what he does is develop scanning protocols to image the brain, particularly brain structural changes in the early stages of dementia. Pretty cool stuff. Here's an overview of what we're going to do today. I'm going to walk us through a lot of steps, kind of like in glycolysis or the citric acid cycle, but again, let's focus on the regulation. So we're going to see that purine synthesis and pyrimidine synthesis happen in two different routes. But in both routes, we're initially forming these as the nucleotide rather than just the free nitrogenous base. We'll see that pyrimidine synthesis is simpler than purine, and you might have guessed that because the structure is more simple. And we'll see how regulation of these pathways is going to tightly balance the levels of the various nucleotides. The last thing I want to do before jumping into the next video where we start talking about the synthesis is remind us of the 6 classes of enzyme. You're going to see a lot of different names of enzyme over these next slides, and some of the names you might see aspects of their class within the name. For example, we're going to see a lot of transferase functions happening here. We might see some hydrolysis. We'll also see definitely an enzyme named lyase, so we can recognize that that's going to do an elimination reaction. And keep these classes of enzyme in the back of your mind as we move forward. On to purine synthesis.